learn to flip these around and express this as probability of response given target stimulus, given the stimulus. Okay, and then our basic approach here is we're going to test for the role of correlations by making two different assumptions for these probability distributions. Either we're going to take the probability of the network response given the stimulus, assuming the cells are independent, or kind of the real distribution, taking into account their correlations induced by the stimulus. Okay, and I just want to point out, again, if we look at the kind of amount of correlation between pairs of cells, it's not very strong. So this is your typical pairwise correlation coefficient, probability of two cells firing together compared to the independent probabilities. Here's the distribution for this experiment, and it's of order 7% or something for the pairwise correlation. Okay, so now let's see what the discrimination error looks like. And so here I'm plotting the error rate versus the number of cells, so we can take subsets of all the cells we reported and calculate do the discrimination, calculate the error, and then in green dots, this is the error for when we assumed independence. And what you can see is, the error drops as you add more cells, but then it kind of saturates. And now in purple, this is the error rate you get when you know the correlations between cells in the population. And so here the error rate drops and drops and drops. And so a couple things I want to point out. When you have this kind of large correlated population, you have extremely low errors. So this is less than 1 in 1,000. So you're kind of getting into the regime where there's actually less than one error in the entire experiment. And there are kind of multiple draws of the population, which is why you don't get 0 as your number. But you're getting into the regime of 0 errors in your 4-hour experiment. So you're really getting to find very, very low error rates for the correlated population. The other thing I want to point out is that, and of course, with much smaller populations, the error rate is really very high. So let's say with one cell, you're getting it wrong almost all the time. And the other thing I want to draw your attention to is the fact that these two curves overlap for smaller populations. So even out to 30 cells, if you completely ignored the correlations that are actually there when you try to read out the visual information, it doesn't make much difference. Okay, so the difference between these curves up to populations of 20 or 30 cells is negligible. Okay, but then when you go to the full population size, the difference in the error is more than a factor of 100. Okay, so basically the effect of the correlation is really weak, not only for pairs, but even for decent-sized groups of cells. But then when you get to the real population size, it's enormously different. Okay, for that analysis that I kind of showed you here, all we're really looking at is whether each cell fires a spike or not. Okay, in kind of a narrow window after the stimulus was on. But if we also look at the time at which that first spike occurs, the quality of the code is much, much better. So this is a similar type of analysis. Now what I'm showing you is the error rate of assuming the correlated decoder versus the error rate for an independent decoder. So unity is right here, and so the correlated decoder has much lower error rates, and a bunch of these are down at 0 errors in the entire experiment. And this is using the entire population. Okay, so the spike latency code is much better performance than kind of spike count, and the correlations also make an even bigger difference for the spike latency code. Okay, so what's the kind of summary here? what I've shown you is that you can make visual discriminations with kind of the ganglion cell population that reach really, really low error rates. Essentially as low as we can measure in a 4-hour experiment. But they require large populations, 100 to 200 cells. Ganglion cell firing patterns are highly redundant. And I think that's related to the high accuracy. And the correlations are critical for kind of reading out this code and making use of the information that is encoded there. Okay, but as I mentioned, we didn't record all these cells simultaneously. So, what we're kind of up to now is scaling up our array. And so we've designed an array that has 252 channels. And so the idea is, a dense array of this size should be big enough to kind of record of order 200 or more cells. So remember, that's kind of the natural size of the ganglion cell population. So we want to be able to record that, in a single experiment simultaneously. So we've gone into the clean room and kind of made these arrays, using photolithography or active ion etching. So here's the whole array, and you zoom in and zoom in. Here are electrodes, and you need to have these. I should put a scale bar here, but this is a 1 micron width to the wire in order to fit them all out of the edge of the array. Okay, so here's just an example of a little snapshot of data from one of these arrays. These are like little spikes if you were to zoom in more closely. And so then of course we want to identify the kind of spike templates. So this is the voltage pattern on the array when each cell fires a spike. So here we're kind of identifying a number of these templates, so they're fairly localized, but you get signals again on multiple electrodes. And then you can take those templates, fit them back to the raw data. So the fit is in red, and the raw data here is in blue. So again, it works quite well. And so this is at the moment really a work in progress. We still want to improve. Some of the arrays have flaws in them, and that really hurts you a lot if you want to record this entire population. So we're improving our process. Another important thing is that the previous spike sorting algorithm that we published a paper on was an algorithm where the human user had to kind of find the templates for each cell. And basically that was fine when you only had 30 electrodes, but now with 252, that's just too laborious. So you're working on kind of more sophisticated automated spike sorting algorithm. I'm not gonna go into a lot of detail here unless you want to ask me about it, but implemented it. We could stand to parallelize more and improve the speed. We do some surrogate tests, and the error rates are very low. So cells that we're identifying look to be quite good, but there's still tweaks we want to make to this algorithm. Okay, so again, this is kind of a work in progress, but the sort of so let me just kind of get back to kind of a big picture view of kind of the population code among